Hello, welcome everyone. Today I'm glad to present our talk. So the presentation is titled CIDO Diagnosis. CIDO means Coronavirus Infectious Disease Ontology. The focus is diagnosis, how we can use the ontology to model, represent, and analyze the diagnosis data. This work is a collaborative work between three people. Besides me, and we have Asia from FDA, and then Dr. Yongqun from the People's Hospital of Guizhou Province, Guizhou in China. Yongqun saw patients of COVID-19, and she's experienced. And Asia is very experienced in modeling and medical informatics. So today I will go with, hold on, let me see. Yeah, a disclaimer first. So this work reflects the views of the authors and should not be construed to represent FDA's views or policies. And I will present some basic background. COVID-19, as we all know, stands for Coronavirus Disease 2019, which is caused by SARS-CoV-2, which is an RNA virus. If you look at the history in the past 20 years, there are two other human coronavirus diseases, which are SARS and MERS. And they actually cause much less deaths in overall. However, the mortality is very high. It's like a 10% or more. So the mortality is higher than the COVID-19. However, their transmission is not as bad as the COVID-19. So as we all know, COVID-19 is getting very badly here now. And here, like from today, June 29th, we have over 10 million cases now and 500,000 deaths overall. So what about the diagnosis? As we all know, diagnosis is a key to control COVID-19. There are many methods of COVID-19 diagnosis, including phenotype identification, lab testing, CT scanning. In the lab testing, like we should mention RT-PCR, which is gold standard, having high specificity, but the sensitivity varies. It depends on many factors, like where you extract the specimen and what time you collected the specimen. And definitely a major issue in diagnosis is how we can improve the sensitivity and the specificity. The COVID-19, the coronavirus infectious disease ontology is basically covering not only COVID-19 but also SARS and MERS. And currently it's being approved to be an OBO library ontology. OBO means open biomedical and biological ontology. It's a consortium. And the CIDO aims to model and represent the diagnosis. Of course, we have also modeled other aspects like etiology, vaccine, drugs, but today we focus on diagnosis. So this is the high-level term hierarchy. CIDO aligns with the basic formal ontology is buffer. So the buffer has two branches. One is continuant, which is a time-independent branch. Another one is occurrent, which is time-dependent branch. So in the occurrent branch, we have process like the RT-PCR SSC or specimen collection process or diagnostic process. And then in the continuant branch, we have material like a specimen, and then we have the disease and the phenotype. and then we have the information. The information can be like the objective of the diagnosis, and then the diagnosis as a conclusion, and then we have the derived data. So in here we have a general modeling. Indeed, we follow the OGMS model for the general diagnosis modeling. OGMS is the ontology for general medical science. In OGMS, diagnosis is defined as the representation of a conclusion of a diagnostic process. So it's basically a conclusion. It's like the data, as you see here, which is conclusion. And then we have the diagnostic process, as you see here as well, is a process. So in the process, we can see it has output of the diagnosis conclusion, but it's based on or has input clinical finding. The finding can be from the phenotype or lab testing or CT testing. So the results from there we can use them for diagnosis. So this slide demonstrates a hierarchical structure of diagnostic methods. So you can see the COVID-19 diagnostic process can be based on different methods, like based on symptoms or chest imaging, which include radiography and CT chest scanning, or serological testing like ELISA, like immunofluorescence serological assay, or viral culture isolation, genome sequencing, or nucleic acid diagnosis. The nucleic acid diagnosis includes different methods, and most commonly we have RT-PCR, and there are also different types of RT-PCR, like a real-time RT-PCR. So next, we are going to ask Asia to presenting the remaining part of the presentation. Asia, please. Asia?
Sorry, I was on mute. Okay, thank you, Oliver. And hi, everybody. My name is Asia Yulin. I currently working at CDRH, FDA, also wear another hat as an affiliated ontologist at NCOR, National Center for Ontological Research at Buffalo. I am going to start with the specimen. The specimen used for diagnosis is an important aspect. For COVID-19 testing, many specimens were collected from different locations in the patient's past airway. CIDO uses OB ontology for biomedical investigations to represent specimen. OB has already existing terms such as nasal swab specimen, nasal pharyngeal swab specimen, bronchial alveolar lavage, or blood serum specimen for us to reuse. And the CIDO team is proposing more COVID-19 related specimen to be included in OB, such as the tracheal aspirate specimen, mid-turbinate nasal swab specimen, and some categorical terms such as upper respiratory specimens. Next. CIDO allows the representation of the sensitivity and specificity of the diagnostic test. Previously, we developed an ontology for biomedical and clinical statistics, OBCS, where the terms are under derived data from inferential statistical analysis can be reused. CIDO created the COVID-19 test sensitivity and COVID-19 specificity under corresponding OBCS terms. Next. In USA, the Americans rely on FDA to evaluate the safety and the effectiveness of medicinal products on the market, such as drugs, diagnostic tests, devices, and vaccines. When a public health threat such as COVID-19 pandemic hits, we are experiencing urgent needs for products to diagnose, treat, or prevent the disease. In such emergencies, FDA can issue an Emergency Use Authorization, or EUA, to provide more timely access to drugs, devices, diagnostic tests, or other critical medical products that may help when there are no adequate, approved, and available options. FDA has issued hundreds of EUAs, including in vitro diagnostic tests, PPEs, related medical devices, and drugs. In this work, we focus on one kind of EUA where the diagnostic tests can be only authorized to use in certain high complexity molecular-based laboratories. Next. An example of such is an diagnostic test developed by Yale New Haven Hospital Lab, which is being authorized via EUA to be only used in the high complexity molecular-based labs in Yale New Haven Hospital. We created a relation called EUA Authorized Use At to link the assay named Yale New Haven Hospital SARS-CoV-2 assay and the location, Clinical Virology Laboratory at Yale New Haven Hospital. The Yale New Haven Hospital is an instance of FDA EUA authorized organization. This data source is from the official EUA summary from FDA's website. Next. We adopted previously developed PCR diagnosis presentation from CIDO brucellosis ontology and provided here a high-level modeling of the test in the context of COVID-19. So this Yale New Haven Hospital SARS-CoV-2 assay is a RT-PCR test, and it is a part of COVID-19 diagnostic processing that is using RT-PCR. The assay applies respiratory specimen and saliva specimen, and it detects both the N1 gene region and N2 gene region that are the part of the SARS-CoV-2 N gene. Next. In conclusion, we presented here the CIDO modeling of the diagnosis aspect of COVID-19, applying existing models adopted from interoperable ontologies such as OGMS, OB, and OBCS. CIDO also represents many diagnosis methods, including RT-PCR. We have to point out that the FDA EUA modeling here, we only models one type of the EUA, that is authorizing the use of high complexity molecular-based laboratory developed test, also LDT. We haven't modeled the commercial kits yet. Modeling the commercial kits, that is an ongoing work and is aligned with activities of OB, LOINC, and the CDC's work of mapping LOINC codes with EUA IVD test kits. Next. So we thank many people's input for this presentation and the work on CIDO diagnosis. Bill Duncan, Severin, Ara Bandi, Ayman Gambala, they are the part of the CIDO team. Cindy Chaudhry, Cynthia Chen, they're from FDA, and Bjorn Peters, he is the PI for OBI, and the other ontology development teams, including BAFO, GMS, OB, OBCS. And Dr. Yu Hong's work is funded by the Chinese Academy of Medical Sciences. Thank you so much for your time and your attention. This is the end of our presentation, and we are happy to take questions. Thank you.
Okay. So we will stop the